Bismillahirrahmanirrahim. In today's presentation, we are going to discuss with you introduction to biochemistry. So, biochemistry is the study of life from a chemical perspective and asks how living organisms arose from biomolecules that are lifeless. These biomolecules can be isolated from organisms and examined individually, and they conform to both the chemical and physical law that govern inanimate matter. A biochemist studies the properties that organisms have that distinguish them from non-living matter. So we are coming to discuss with you diversity and speed of life. So inside of any given living organism or cell, all living organisms are composed of cells as we know, so they have a high degree of chemical complexity and microscopic organization. Their intricate internal structure have characteristic sequence of subunits arranged to produce, for example, protein with unique three-dimensional structure. Each of these unique protein is highly specific for what types of molecule it will bind and interact with. Living organisms also have system for extracting energy from the environment as well as transforming that energy into a usable form. They possess capabilities to replicate their genetic information and reproduce components of the cell, as well as the capacity to precisely regulate the interaction between those components. This sophistication applies to the microscopic structure, for example, the heart, but also applies to microscopic structure and individual chemical compound throughout the organism, and the world outside of it come into contact with each other inevitably. This requires cell, tissue, organ, and organ system to coordinate and compensate for exchanges in another. Possessing these abilities allow the whole organism to be greater than the sum of its individual parts. On the microscopic scale, a biochemist sees a collection of molecules carrying out a program whose result in both program reproduction as well as the cell perpetuation of that collection of individual molecules. In short, the biochemist sees life. So this is a system of life diagram. So we are going to discuss history, philosophical and technological environment improvement. Throughout history, organisms have changed their strategies to adapt to the changing circumstances on our planet. These changes have passed through inheritance over eons, and the result of this is the enormous diversity that has evolved. On the outside, a human being looks very different from a mushroom, but fundamentally they are both related to each other through their shared ancestry. The human and the mushroom do not share every common property, but they do share a common chemical framework. This chemical framework is what evolved to allow different organisms to survive in a vast array of habitats, from insect animal intestine to thermal hot spring from 5,000 feet under the ocean surface to the coffee shop where you might be studying today. Living known living organisms fall into one of the three large domains that define the branches of the so-called evolutionary trees that show their course from a common progenitor. There are two groups of prokaryotes that can be distinguished for biochemistry reasons: the archaeobacteria and eubacteria. Archaeobacteria usually live in extreme environment, whereas eubacteria such as E. coli live in the same environment as eukaryotes do. Biochemistry, philosophy, and technology have both co-evolved throughout their course history. Scientists have been asking biochemistry-related questions for hundreds of years with no definitive start date. The French nobleman Antonio Lavoisier is known as the father of modern chemistry, but he began asking some of the first questions of biochemistry in his 1777 study of how combustion is tied to respiration. Diastase, now called amylase, was discovered by a French chemist named Anselme Fane in 1833. but most consider the birth of biochemistry as the field of study to have begun with a German chemist named as Edward Bucher described the process of fermentation in 1907. The mid-20th century marked an explosion of the technique development for the field of biochemistry, such as chromatography, X-ray diffraction, NMR, radioscope, labeling, and electron microscopy. These tools allowed scientists to analyze individual molecules and protein and deduce metabolic pathway. American biochemist Corley Mullis won the Nobel Prize in Chemistry along with Canadian biochemist Michael Smith for improving the technique of applying DNA through the polymerase chain reaction PCR, first described by the 1968 Nobel laureate H. Gobind Khorana from India. The philosophical view of biochemistry have also changed throughout history. The ideas leading to the hypothesis of vitalism began in the ancient Egypt, where state living organisms are fundamentally different from non-living entities because they contain some non-physical elements or are governed by different principles that are animate things. This idea was widely accepted by many, including Swedish chemist and physician John Jacob Berzelius, who in the early 19th century discovered several of the elements on the periodic table. So this is DNA animation diagram. Ultimately, Friedrich Wöhler disproved this hypothesis by synthesizing urea, a constituent of urine, from ammonium and cyanide in the laboratory without the use of a cell. Philosophical way of biochemistry have co-evolved the development of technology. For example, Friedrich Meister isolation of DNA in the early 1900s. The Avery McCourt experiment demonstrated in 1944 that DNA is the substances that cause bacterial transformation, leading to the 1953 determination of DNA structure by Francis Watson and James Crick using X-ray diffraction. This experimental evidence allowed for the development of the central dogma of molecular biology in the 1960s, which states that genetics information flows from DNA to RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and then to protein.